This is the first of the preps for lecture 17, and we're going to be looking at single gene traits in this situation. And we'll be looking at a lot of different complications. So not every set of alleles has just one dominant, one recessive. The dominant alleles aren't always completely dominant over the recessive. So on these are sometimes referred to as non-Mendelian genes, which I think is just wrong, because the genes are still segregating the way Mendel, described the segregation. It's just that there are now cases where there's interactions. There are multiple alleles. Sometimes the dominant allele isn't completely dominant, and sometimes both alleles are functional and contribute to the phenotype. So there's a lot of different complications, let's say, of these kinds of situations. So we're going to be looking at sort of, dominant recessive alleles that are variable in terms of how the dominance part works, variations and so on. We'll look at conditional alleles. We'll look at what we call an allelic series. So you have a particular gene. There are a bunch of different alleles of those genes, and you can get variations in the phenotypes based on the relationship between one allele being dominant over another, for instance. And anytime you have a chance to put up pictures of cats, it's, that's a good opportunity. So we're going to start with the simplest of the variations in dominance. And this one is called incomplete dominance. And so what this means is that you have two different alleles. One is producing a functional enzyme, a functional protein, and the other is not producing a functional protein. However, in the organism, there is, you would call it a dose, a required dosage. So heterozygous individuals have one functional copy and one non-functional copy of the gene. So they only have half of the protein's activity, whatever that protein is doing. And so this is a case of, there's a number of different plants that have this kind of situation. You have, if you have pure breeding red flowered plants and you cross them to pure breeding white flowered plants, when you produce heterozygotes, the heterozygotes have an intermediate phenotype. And so this is called incomplete dominance because the functional allele is not completely dominant it doesn't completely compensate for the problem with the recessive allele. Okay, and so what's really useful in a way about this, if you're a geneticist, is that when you're looking at progeny from crosses and so on, you have no need of this kind of notation with the dash, because we know the exact genotype based on the phenotype, because the heterozygote has a unique phenotype compared to the two parents. It's intermediate between. And the way I think about this, if I'm trying to imagine it, I think of buckets of paint or something like that. So you've got the dominant allele. It makes an enzyme that is involved in making red pigment. So it makes a bucket of red paint, say. And then you have the recessive allele, and it in fact doesn't make any paint at all. So if you have two dominant alleles, you have two buckets of paint. So you paint the wall and it's nice and dark red. If you are heterozygous, you only have one bucket of paint, but you still have the same space to spread it across. And so that gives you a lighter shade, an intermediate shade. And of course, if you're homozygous for the recessive, you have no buckets of paint, and therefore you have no red pigment. So a lot of people talk about the white pigment, but generally when you see a white flower or a white organism, It's a lack of pigment. It's not that there's white color being put into the flower, it's that there's no pigment available. And so it's kind of empty in a sense. So this paint bucket kind of analogy may or may not make sense to you, but I would encourage you when you're thinking about this to try to make some sort of analogy or picture that helps you understand what it means. I do find for me analogies that are really tangible, thinking of like the bucket of paint or the paintbrush or whatever. For me, that really helps me remember and understand things, but for you, that might not work. So you should look for something that's going to actually work for you. So here's the second one to contrast. It's called codominance. And what this means is that neither of the alleles is dominant over the other. They both contribute something to the phenotype of the heterozygote. But the heterozygote has a phenotype that is distinct from, it's different than either of the homozygotes. And so it's not just an intermediate, like a paler shade of red. In fact, the codominant phenotype is distinct. It's different. And the famous example of this is human blood type. I'm going to go through the ABO blood type, but you should know, and we'll probably talk about this later in the semester, there's also a blood type called MN, and we're not as familiar with it because we don't have, for example, the huge immune reactions and things that, that we know of with the ABO blood type. You may know that certain people can donate to anybody, but can only receive blood in a donation from people that have the same blood type as them. And there doesn't seem to be as much of a problem with the MN type. But I will go over it later in the semester because we'll be using it as a way of looking at population genetics. And so in this case, the molecular basis of the ABO blood type is known. And when we go through it, you'll see why this is codominance. Okay, so the gene is called the I gene, and you can have the A allele. So you would have 2 copies of the A allele, and that would be type A, or you can have 1 copy of the A allele and 1 of the O allele, and that would also make you type A. So this is just a normal complete dominant situation. Same with blood type B. You can be homozygous for the B allele, or you can be heterozygous for the B allele with the O allele. And that makes you type B. So both of these are just examples of normal complete dominance, the same kind of things that we've seen before, except the notation is just a little bit different. If you're homozygous for the recessive allele, little I, you have type O blood. And I'll explain the molecular side of this in a minute. But if you have 1 A allele and 1 B allele, you have type AB allele. And this is called codominance because each of these alleles is contributing something to the phenotype, and it can be distinguished from either of these 2 parents. And it turns out that it is, if you look on the surface of the blood cells, the type A allele has a protein on it with a certain shape. The type B allele has a protein with a different shape. And so when people are type AB, their blood cells have both of those shapes on there. And so it's not intermediate, it's in fact different because the actual proteins that are on the surface of the cells differ. And you'll see a picture in a minute. So both of these 2 alleles they're both functional. They encode enzymes that attach these different shaped molecules onto the proteins on the cell surface. And so this is how it works. It's the sugars that are being added onto the molecules on the surface of the blood cells. And the reason why these are very important is these are some of this isn't all of them, but these are some of the proteins that are used by the body to monitor which cells belong, which are self, which cells are belonging to the body, and which cells are potentially invading pathogens. So the body knows, if you're type A, that these molecules on the cell surface are supposed to be there, and they are their self. And the enzyme that's encoded by the A allele puts a certain set of glycoproteins, so sugar molecules, in a certain shape on those proteins. And so the body can recognize that. If a person is type B, the sugar molecules are placed on the same protein, but they're placed in different positions. And so you've got this slightly different shape. And if you have type AB blood, your blood cells have both of these types on them. And so you can see the shape and the form of the cells are slightly different. If you are type O, you don't have any functional enzymes. And so those sugar molecules are not put on the surfaces of the cells. The consequence of this is that people who have type AB blood, if somebody with type B blood donates blood to them, their immune system can recognize these molecules on the surface and go, oh yeah, this is all right, no problem here. And the same with type A. If type A people donate blood, then the immune system of the AB person can recognize those. Oh yeah, that, we know that's supposed to be here. We have that on our cells too. So they can accept, and they can accept, of course, blood from type O because there are none of those sugar molecules on the outside. Okay. And so people with type AB blood are considered the universal acceptors because anybody can donate blood to them and they won't have a serious immune reaction. However, if I'm type O, and so I can donate my blood to anybody and it won't cause any reaction, but I cannot receive blood from any of these other types because my immune system will look at these and go crazy, and it will start attacking the blood cells that have the wrong molecules on the surface because the immune system is responding to, it's kind of like stranger danger. It's like that would be a bacterial or a virus cell, and the immune system wants to get rid of that. It's not supposed to be there. But sometimes the immune system overreacts and will start attacking also the normal blood cells too. And that's why you hear these terrible, a long time ago, we didn't have really good cross-matching, and so sometimes people could die from a blood transfusion. And so these days, before surgery, you can go and donate some of your own blood so that if you need blood after the surgery, it can be used, and then for sure it's going to be the right type. Anyhow, so hopefully you can see though why this is codominant, because on the surface of these cells you have both of the antigens, some of them placed there by the enzyme from the A allele and some of them placed there by the enzyme of the B allele. Okay, and so this is distinct from either of these two. So codominance and incomplete dominance relate to single genes, and sometimes when I am doing questions around two genes, sometimes, especially after the next few lectures when we talk about two gene interactions, students will sometimes say that one gene is dominant over another, and we have a completely different term for that. So when we're talking about dominance, whether it's complete dominance, incomplete, or codominance, we're always talking about particular alleles of single genes. Okay, and for both of these cases, you can use the phenotype to determine the genotype, which makes things like pedigree analysis, or especially if you're doing some complicated set of crosses or something, you might want to consider what the advantage might be of having codominant or incomplete dominant alleles. Okay, so that's the first little bit. Now I want to talk about, this is a more common situation, which is that most genes have more than just two alleles. So we don't just have big A and small a, although that's a really good construct for us to begin to learn, first of all, just how the genes segregate and how dominance actually works. But a more realistic picture is a number of different alleles known of a particular gene, and we often superscript them. So we will often have small a with like plus, that's your wild type allele. And then we often have superscripted either numbers or sometimes letters. You'll see an example coming up of a case where the letters indicate something about the phenotype of those alleles. Now, some of these alleles might be more severe than others. They might have what we call strong alleles, they have a severe phenotype. They're very different from the wild type. Think of flower pigmentation like the 4 o'clock flower. A severe mutant will have no pigment at all, or just a slight tinge of pigment in the petals, whereas a weaker allele might have a more moderate or a milder phenotype. A weak allele is closer to being wild type. So, for instance, if you have coat color in cats and the coat is supposed to be black, then a weak allele might make it a dark gray color, whereas a strong allele might make it like very pale gray or even white. So when we're talking about the strength of the alleles, we're actually talking about how different they are from wild type. A weak allele is not much of a change. It's the protein is mostly doing the job okay, but maybe not quite up to snuff. And so the phenotype is closer to the wild type, and the phenotype is not what we'd call a really serious phenotype. Strong alleles have a very severe phenotype. They're really messed up compared to wild type, or they're very, very different from wild type. We can see a big difference, okay? And then when we're thinking about the dominance interactions, I really want to emphasize this because it's sort of counterintuitive, because usually the weaker alleles show dominance over the stronger alleles. And that doesn't sound right at all. But that's because the weak alleles are more like wild type. So you have an allele that's producing a protein that's mostly doing the job. And the strong allele is more abnormal. Maybe the protein isn't doing anything at all. And if you combine a protein that's partly functional with one that's non-functional, the one that's partly functional kind of wins. Okay, and so we have an allelic series from the weakest to the strongest alleles, and then the greater than sign, greater than means dominant to. So if we were combining A1 with A4, for example, then the individuals would probably look like A1. If we combined A4 with A3 here, then the individuals would look like A4, because the A4 is a weaker allele and it's going to cover the effect of A3 and so on. Some other things to think about is sometimes these superscripts are just letters, and I'm going to show you an example of a couple of those in the next prep. But just also note that the allele numbers are generally listed in the order in which they were discovered or characterized, and so it doesn't have anything to do with their dominance. So A1 isn't always dominant over everything else. It may happen to be in one situation, but A1 could be the weakest or the strongest allele, in another case. That's just historical. That's the order in which they were found or characterized or understood, and it doesn't have anything to do with dominance. So think about this a little bit, and then in the next prep, I'm going to go through a couple of examples.